Hello everyone, and welcome to today's webinar, Cell Culture Bioprocess Scale-Up Workflow from Bench to Pilot Production Scale using Single-Use Bioreactors. I'm Alwin Albers of Eppendorf, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Eppendorf. Eppendorf AG is a leading company in the life sciences that develops and sells instruments, consumables, and services for liquid handling, sample handling, and cell handling. To learn more, please visit www.eppendorf.com. The webinar will be around 30 minutes, and we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we'll have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing educational credits. Please follow the process to obtain your credits by clicking on the continuing educational credits window. I'd like to welcome now our three speakers who are going to address different aspects. First, Marcia, the head of Bioprocess applications at Eppendorf, Rich Mirro, business manager Bioprocess at Eppendorf, and as the last person, Amanda Suttle, research scientist at Eppendorf. With that, I hand over to Ma. You may now begin your presentation.
Thanks, Alvin. Presentation today will have three sections, as Alvin had pointed out. So I will be starting it with the ScaleUp Strategies, and followed by the Bioflow 720 introduction. And then Amanda will talk about the workflow of involving the 720 using the ScaleUp Strategies. As Alvin mentioned, I am Masha. I manage the Eppendorf Bioprocess Applications Labs. We have a lab in Enfield, Connecticut, USA, and another applications lab in Jülich, Germany. And this work on the 720 scale-up lab work is conducted in Enfield. So let's start with why scale-up. The purpose of the cell culture scale-up for most people is to bridge the gap between the R&D and production. Typically, scientists use bench scale, for example, or small scale for optimization work, and until they get really good yield. And to bring that yield into production scale, it is not a trivial task. So this scale-up effort often involves the intermediate step for the pilot scale that goes from 50 to 2,000 liters. And the introduction of the 720 controller is for managing the pilot scale. I'll start with introducing some of the scale-up strategies we investigated in the applications lab. One of them is the constant mixing time. It is a popular strategy from the chemical engineering field, and which precedes to the bioprocess business. And when we looked into the constant mixing time, we realized that it wasn't actually practical enough, because the small scale tends to have extremely fast mixing time, and in many cases actually does not overlap with a bench scale or a pilot scale. So we weren't able to find constant mixing time to implement this particular strategy. The next one, which we do use, and it's fairly straightforward, is constant tip speed. The constant tip speed maintains a constant shearing force for the cells. So for cell culture, at least you're not subjecting the cells at a different shear force or a little harsher conditions on a larger scale. The method does work when the scale are not too far apart, especially popular for smaller companies where exact yield matching isn't critical. And to the right, we have one application note where we use the constant tip speed. to scale from 0.25 liter to 1 liter. Those are small scale. So there's a barcode that points you to the actual application node. The next scale-up strategy is a really popular one, the constant KLA. What we consider as a former gold standard, a constant KLA for anaerobic cell culture, anaerobic bioreactor culture process, will give you a constant yield. The difficult part is the KLA tends to change with most of the parameters used in culture, whether it's agitation speed or amount of gassing. So that means to match a KLA between one scale to another, we would have to analyze many, many KLA parameters. And that is kind of tedious. We do publish KLA numbers on selected vessels. However, our scale-up strategies that we've been practicing is the next one, constant power over volume, constant PV. This is what we consider as the current gold standard for the biological scale-up. The big advantage is simplicity. One would only need to measure one power number per vessel, and it doesn't really change based on the vessel conditions. And with the power number, which is typically measured by torque, impeller torque, you can see the picture to the right. We have this apparatus over on top of the vessel. The little red piece in the middle is the torque sensor. Then there are two adapters, flexible adapters, one above and one below. Adapt the torque sensor to the shaft, which measures impeller torque through this setup. And the power number is measured one per vessel. And the disadvantage is this particular method is not something that everybody already have in place. If you measure KLA, you don't need any of these. Anybody can do it. But to measure the power numbers, you kind of need to set up this sort of customized apparatus. So here is an example. We have released the power number for all the BioBlue vessels. And the barcode links to the presentation where this particular power number sets are released. It was actually released into a application where we did a scale-up of chill culture in the BioBlue. So in the next slide, you can see with the power number, then one would calculate the power over volume. And when we plot the PV over different tip speed on different vessels, this will allow you to find your parameters for constant PV scale-up. On the right-hand side, you can see that we put into a sort of two line indicating the scale-up zone. In this particular range of agitation and the vessel chosen, you can look for overlapping power numbers to conduct a constant PV, overlapping PV to conduct constant PV scale-up. And this zone changes when you change or increase your agitation speed. So on the left-hand side, the figure actually shows we go up to about 1 meter per second of tip speed. The increase the tip speed allows a scale-up to be done at a higher PV levels. So the next one is an example. When we apply the constant PV scale-up strategy to the three selected vessels shown, 0.3C, 3C, and 50C, and we were able to get really nice cell culture yield matching. So the picture on the left shows the cell growth profile between the three different vessels with no optimization other than maintain the PV being constant. And to the right is the antibody production yield. This is a chill cell line producing a human monoclonal antibody. So on the right-hand side, you can see the antibody production yield was maintained very nicely. So these strategies are used in later on, you will see the presentation. These strategies are used in our ScaleUp using the new Bioflow 720. So that concludes my section. I will pass the presentation to Rich Mirro.
Okay. Thank you, Ma, and hello to everyone. Thank you for joining us today. My name is Rich Mirro. I'm the business manager at Eppendorf, responsible for bench and pilot production systems. Today, I'll be providing a brief introduction to the Bioflow 720. So as we all know, bioprocess facilities face many challenges today. There's resource limitations, limited space, and often the challenge of working with or using products from multiple suppliers. And to help alleviate these challenges and others, I'm proud to introduce the Bioflow 720. Designed based on 4 basic principles: scalability, efficiency, flexibility, and application-driven. The new product features hardware and software designed to facilitate workflow and alleviate the challenges mentioned, which are commonly found in bioprocess suites. I look at the first principle, scalability. As you can see from the illustration, the Bioflow 720 adds additional scalability to our already strong portfolio of instruments, ranging from small parallel systems through bench and now pilot production. This supports a workflow from R&D to production for our customers. The system is compatible with the Thermo Scientific High-Performa 5:1 single-use bioreactor system with volumes of 50 liter to 2,000 liter. And when you combine that with the existing Eppendorf BioBlue single-use portfolio, working volume ranges go from 100 milliliters up to 2,000 liters total. I think very impressive working volume, covers most applications. Further supporting the element of scalability, the advanced software features integrated tools which simplify tasks. As Ma had mentioned, we have a new tool called the ScaleUp Assist, which allows for quick and easy scale-up models to be developed based on a variety of strategies: constant power, constant tip speed, and a constant gas flow. During the third part of the webinar, Amanda will go over the tool in more detail, and she'll provide some examples of how this has been used in our laboratory. If I switch to the second principle, efficiency, the new software and instrument features additional software, auto-calibrate and auto-inflate, both of which are designed to save considerable time by automating repetitive tasks. The auto-calibrate polarizes, zeros, and then spans DO sensors based on a repeatable computer logic. The screenshot you can see on the right is an example of the auto-calibrate, and the workflow for this, users would select whether they want to calibrate a single or all DO sensors. They would select whether or not the calibration will be done inside or outside of a vessel, select the 0-point strategy, electronic or nitrogen, and then select key variables like gas flow rate, agitation, and temperature, as well as polarization time. Simply press play, and the system automates the process, which can save up to 8 hours if you're looking at an analog DO sensor, which requires 6 to 8 hours of polarization. So not only does it save time, but it also creates a repeatable DO calibration. Operators may select span or 0 at an individual preference of time. This process automates it and does it the same way every time. The second feature, the auto-inflate, automatically adds gas to the bioprocess container in 2 stages. After the first stage, the bag is inflated approximately 50%. There's a pause. The bag can be adjusted to make sure that the impellers, that the sensors and other critical components are seated properly. Then the process is continued, which fills to 100%, preparing for media to be added. There are safety interlocks built in. This way, if an exhaust filter or any other blockage occurs, the process stops if pressure is at or exceeds a specific set point. Additionally, with efficiency, the software is built and designed using the same user interface concepts as our existing Bioflow 320 and 120 products. And this really simplifies the transition for existing customers as well as customers who may be looking at products for both bench, small scale, or R&D, pilot production. In the screenshot examples on the left, you see an existing screen for the Bioflow 320, and then you see the same screen on the right-hand side for the Bioflow 720. In principle, they're almost identical. There's some new features added to the Bioflow 720, but the biggest adaptation was moving from a 4x3 format to a 16x9. Looking at the third principle, flexibility, the general Eppendorf design philosophy provides standard systems with configurable options. And this allows us to design a standard control system and meet the benefits of standard design practices while enabling the ability to configure it in thousands, actually, of different permutations. A couple examples, the highly configurable gas flow options. So up to 5 configurable TMFCs, thermal mass flow controllers, can be selected for the overlay, the sparge, an optional second sparge, or a CO2 stripping. And those TMFCs can be configured and mixed and matched at different flows to meet the preference or the demands of the specific application or process. Also, because of the digital nature of those TMFCs, they can be easily exchanged in the future if the instrument is needed for different campaigns or different purposes. From the perspective of sensors, up to 4 sensor connections can be equipped on the instrument, and they're compatible with both analog, ISM, Mettler-Toledo, or digital, as well as the Hamilton sensors. There are several pump options. 3 Watson-Marlow 314 pumps come standard, but additional Watson-Marlow 520 pumps can be added, as well as additional 620 pumps could be plugged in for high flow requirements. Looking at the fourth and final principle, application-driven, the product is truly designed for bioprocess. One of the elements we show here is the mobile control station. Allows the instrument to be quickly moved from laboratory or suite to suite if required. That allows for quick deployment into a variety of campaigns and a lot of flexibility. The overall footprint of the controller is quite narrow, 32 inches or 81 centimeters. Takes up very little lab space. The user access panel, shown on the instrument on the left-hand side, closed in the image, streamlines, and it protects sensitive electronics. In the previous screen, you could see it open where an operator was connecting sensors. That panel allows cables to be routed through an integrated cable management system, and out the bottom, it allows for both organization and protection and ease of cleaning overall for the system. So those are the 4 key principles of the new product. Please let me know if you have any questions later. Amanda.
All right, thank you, Rich. My name is Amanda Suttle. I work in the applications lab in Enfield. And today we'll be presenting our ScaleUp work, integrating our newest bioprocess family member, the Bioflow 720. So here we have a typical bioprocess antibody production workflow overview. Our workflow begins when we thaw our cells, and this can typically take less than a day between our preparations and getting our cells into their incubator. When we move on to the routine passaging and flask culture scale-up, that can depend on the inoculation density you're trying to reach for how long you're in that phase of the workflow, which can be between 2 to 3 weeks of culturing typically. For our study, our routine passaging and seed train scale-up was about 2 weeks. And lastly, we have our bioreactor. So again, the stage can vary depending on the user process. For example, a typical batch run runs for about a week. A fed batch can take anywhere from 2 to 3 weeks, and a perfusion can last 3 or more weeks. For this study, we chose to run batch runs. When designing our scale-up process, it is critical to achieve matching growth curves and production yields from shaker to pilot scale production. So what's our secret for having robust inoculum? That would be the New Brunswick S41i, which is pictured here on the right. It combines precise temperature and CO2 control with one of the best laboratory shakers available, resulting in high cell yields and viability. The S41i has a wide range of interchangeable dedicated platforms for various size flasks, and it is easy to change between experiments, allowing it to flexibly adapt to your process. It also has one of the most important features, my favorite feature, the high temp disinfection. With a few simple clicks, your shaking incubator is sparkling clean and ready to go for your next bioprocess adventure. So why are constant parameters important to any experiment, especially scale-up? Keeping constant parameters gives consistent results, which means really reliable data. Constant parameters also reduce shock factors to the cells that could potentially have a negative effect on your culture, like exhibiting a lag later on in your bioreactor run. In addition, when keeping scale-up processes in mind, it is extremely important to keep constant parameters and reduce those shock factors to achieve the matching growth curves from small scale all the way up to our pilot scale. So what is our main goal for achieving perfect inoculation? We want to keep a viability 95% or higher in our pre-bioreactor culture so that our cells remain happy and healthy when we put them into our bioreactor. So how do we do this? We track initial flask culture in an Excel file, and this file includes past data so that we can compare our current data to get an idea of where our cells are tracking in growth and viability. This is extremely important so that we can quickly detect any outliers or unforeseen variables. For instance, we take a cell count every day and look at our data individually and how they compare to other flasks that we're culturing. But the problem with doing only this is that we're not able to see any changes over time, and everything might look okay from one day to the next, but compared against other data, it might not actually be. And we don't want to inoculate our bioreactor with subpar cultures. Something else that has helped our CHO culture was finding an optimal passage schedule for our cells. It seems simple, but is very important. If your cells are passaged too much, it can lead to unhappy cells. If they're passaged too little, ammonia can build up and cause adverse effects to your cells. And in the initial phases of development work, this is really important to uncover. We found that our CHO cells like to be passaged every other day for optimal growth. Next, we monitor our pH and keep tight parameters on them. This is especially true when it comes time to inoculation of our bioreactor. We want to make sure that the bioreactor culture medium and our cells flasks that are at the same pH, we take offline samples to compare and set our bioreactor pH loop set points accordingly so that they are not shocked in their new environment. And lastly, we try to keep our cells inside their optimal growing environment, aka the incubator, as much as possible. We prepare everything we need to passage our cells ahead of time inside our biosafety cabinet. Temperature and pH can change drastically outside the incubator, so we try to, like I said, cut down on that. And also, this also allows, for settling to occur. And our CHO cells are already prone to clumping, so we don't want to, have them out too long. By following these steps, we've acquired the perfect inoculation for our bioreactors with happy, healthy cells. So now that we have the robust inoculum, how do we integrate the ScaleUp Assist into our process? So as Ma has already mentioned, we've touched base on some of the great ScaleUp strategies used in the industry today and the logic behind them. So what strategies does ScaleUp Assist support, and how do we choose, or what did we choose, for our study? ScaleUp Assist will allow you to use a constant tip speed. It also supports maintaining a proportional max gas flow. And lastly, it allows you to maintain a constant PV. And for this study, we chose to use constant PV as our ScaleUp strategy. So here is our ScaleUp Assist screen. And I'm just going to kind of take you through how we inputted information to get to where we had our parameters. So first we selected our ScaleUp strategy, which is located at the top. So as you can see that the PV is selected. We then selected our first vessel. In this case, we chose the BioBlue 3C. We then chose a 10C and a 50-liter sub for our other two vessels, as you can see in this screen. Once we selected our vessels, all fields for the ScaleUp parameters will be auto-filled with values. And this is just something that pops up. And once you start changing values, it'll recalculate. And because the vessels are already selected that we selected are already integrated into the software, ScaleUp Assist has already done all the work for us. As you can see, you have the impeller diameter and the power number already in there, and it's not selectable because that is already something that won't change. So next, we select our PV box and input a value of 20. That's what we chose for this study. We then chose our working volumes for all vessels. For the 3C, we chose to run a working volume of 3 liters. For the 10C, we chose 10 liters. And for the 50-liter sub, we chose a working volume of 40 liters. Lastly, we selected a maximum gas flow rate of 1 SLPM for our BioBlue 3C, and it auto-populated the rest of the parameters needed for a ScaleUp process for all 3 vessels, like agitation and max gas flows. So as I just mentioned, we chose our 3C, 10C, and the Thermo Scientific High Performa 50-liter bioprocess container. I've already mentioned the working volumes. Our agitation, like I had mentioned, was generated from the ScaleUp Assist software. So for our 3C, it generated 151 RPM. For 10C, it was 126 RPM. And for our 50-liter sub, it was 169 RPM. Our target inoculation for all experiments was 500,000 cells per mil. Our temperature was set to 37 degrees C and kept constant for the duration of all of our experiments. We used ISM-DO sensors at a set point of 50%. We used a gel-filled pH sensor set to 7 with a dead band of 0.1. And this was controlled by cascade to CO2, which was our acid, and cascade to 0.45 molar sodium bicarbonate, which was our base. We used 3-gas auto mixing and set our gassing range accordingly based on our ScaleUp Assist parameters. We also always like to set a minimum flow, which is usually the lowest our TMFC will go. In this case, it was 0.04 SLPM. And this is just to avoid any backflow through our sparge filter since demand is so low at the beginning of a run. I wanted to highlight our vessel preparations for our 50-liter sub because it's a little bit different than our regular BioBlue single-use vessels. So first we unpacked and inspected our bioprocess container. We connected an aseptic pressure sensor to our bioprocess container, which is needed to use the auto inflate and also to detect any pressure within our bioprocess container while running it. We then carefully placed it into the support container, which is pictured to the right. We use the auto inflate feature, which is automatically introducing a specified amount of air into the bioprocess container until it's fully inflated, like Rich had mentioned. And it also has a built-in safety feature, as he also mentioned, that will shut down all gases in the event of high pressure being detected. So that's really great. After the bioprocess container has been properly inflated, we insert our sensors aseptically by using a sub-probe assembly that has been previously sterilized by autoclave. And we wanted to do that before we put any media in, because as you can see on the right-hand side, the sensors are at the bottom. So if we were to do that with the liquid, it would just be a lot harder. So we always do it before we put any kind of media in there. Then we filled our bioprocess container by weight using the load cell. However, the Bioflow 720 has a bolus feed addition feature that can make adding media simple. And as Rich had also mentioned, there's also an auto calibration for DO sensors, which saves time and calibration, makes calibration reproducible between users, which is really important. For our 50-liter sub run, we needed a larger volume of inoculation than our previous BioBlue cultures. So we first started our culture using an S41i. We then inoculated a BioBlue 10C with a working volume of 10 liters once we had enough cells. Then we grew our culture for a few days in our BioBlue until it reached approximately 2 million cells per mil. And then we welded onto our harvest line on that vessel and inoculated our 50-liter sub. So in this diagram, we show our scale-up work that we have successfully completed using the blue arrows. Our scale-up flask culture strategy that we use for our 50-liter sub run can also be applied to larger vessel sizes. For example, if we wanted to inoculate a 250-liter vessel, we would first prepare our flask culture, like I had mentioned before, in our S41i. We then scale up to 2 of our BioBlue 10Cs to allow the culture to grow up for a few days to achieve the calculated density needed to inoculate our 250-liter sub. And the same logic could also be applied to a 500-liter sub running the 50-liter sub as a seed train to achieve enough culture to inoculate the 500-liter sub. Throughout our experiments, we monitor our metabolic profiles twice daily to observe how our culture is growing and to quickly detect if anything is wrong in our culture. So here we have our metabolic profile of our 50-liter sub run. Our actively growing culture depleted the initial supplied glucose by day 5. Lactate remained under 2.5 grams per liter for the duration of the run, and ammonia concentrations rose every day to a toxic level of 10.2 millimoles per liter by day 6. We reached a peak antibody production by day 6 of 164 mg per liter. And here we have our cell growth curves and antibody productions for all 3 vessel sizes. And as you can see, they're very similar. It demonstrated that the new ScaleUp Assist feature can decrease the process development time while maintaining cell yields across various sizes and bioreactor platforms. So in summary, choosing an appropriate strategy is critical for a successful scale-up of a cell culture. The Bioflow 720 ScaleUp Assist software can, the features simplify calculations necessary for scaling relevant parameters. And finally, we've demonstrated the possibility, of matching yields for 3-liter to 50-liter sub bioreactor culture. And to find out more, you can scan our QR code for our application note regarding our cell culture scale-up workflow. And at this time, I'll give it back to Alwyn, and we will take some questions.
Well, thank you, Amanda, Rich, and Ma, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question that we will address is that the seed train, why did you show multiple BioBlue 10C runs and not more runs in the BioBlue 3C or 50C, 50 liter? Amanda, please.
Okay. So we did more runs using the BioBlue 10C because we knew we were going to need a higher inoculation density for a 50-liter subculture. So we wanted to figure out how fast our cells were growing in order to figure out the best time to harvest our cells. And as we mentioned, that we found that the correct density for us for this run was around 2 million cells per mil. And that's basically why we have extra runs for our 10C.
Okay, thank you. Another question that we have, do we have or are the power numbers of stainless steel glass vessels released for 1 liter and 10 liter? Not sure who can answer that best.
I could answer that, Alwyn. This is Rich Mirro. Within the ScaleUp Assist autoclavable vessels for Bioflow 320, 1 liter, 3 liter, 5 liter, and 10 liter, the water jacketed version are included.
Okay. How quickly does the software trigger or respond to a drift from ideal settings of temperature, DO, or pH?
I could also answer that, Alwyn. Those loops are controlled using a PID response. Some can also be set up with a deadband. So to answer the question, as soon as the present value drifts from the set point, the instrument will begin to control to try to correct the error. But that could also be tempered by introducing a deadband if you don't want instantaneous control or correction of by adding a base, for example.
All right, thank you, Rich. A question on the ScaleUp Assist software. Is it only offered on the Bioflow 720 controller, or is it standalone software that we could use to scale the 50-liter Cellogen Blue to the high performance or other places? Is it just scaling or just calculating P over V and VVM for you?
So if I understand the question correctly, I'll try to answer. The software is integrated into the Bioflow 720, so it is only available currently as a feature of that product. We are looking at the possibility of integrating it into our SCADA platforms, but that would be for future release, not today. And then to answer the second half of the question, it's calculating the recommended P to V or the tip speed based on the initial parameter set for sort of what I call the driving vessel, the first column. And that could be done for either scale up or scale down. So some applications may call that you want to know from a 500 liter what it may be at 50 liter and then down to a 1 liter or whatever your scale down. So that could be done in either direction.
Okay, thank you, Rich. Can the pilot system handle cell culture with microbeads?
So I guess I'll answer that also, if Ma doesn't mind. So there are application notes available from Thermo Fisher Scientific supporting the use of microcarrier beads within the Thermo High Performa 5:1 sub. So the product is capable of it. The instrument, the Bioflow 720, could of course support that application.
Maybe an extended question there. Has the process been used in the production of cell exosomes?
So I don't have record of any documentation. Ma, perhaps, would you be able to answer that?
I can do a partial answer. The exosome, especially from stem cells, is fairly new. And from our bench scale, we just actually started doing some applications for it. So we have one application node just came out that was using a bench scale for mesenchymal stem cell exosome production. We've not looked into something at the a pilot scale, We have another application coming up that is going to be the same application, but using the DOS box. So at this point, the information that we have is limited to bench and small scale. But in the future, if the exosome market becomes substantial, we will probably look into it. But right now, we don't have anything in the pilot scale.
Thank you, Ma. The seed train showed, I think this would be for Amanda, this question, would it also work to inoculate a 2,000 liter culture?
Yes, I can answer that. Yeah, using the seed train strategy that we've come up with, certainly you can adapt it to your process accordingly to figure out how much inoculation density you need to get to the 200 or 2,000 liter rather. But basically, kind of like how I said, for a 500 liter, maybe you would use the 50 liter as a part of the scale up to grow up enough cells and inoculation density into your 500 liter. So you'd have to take it as that type of example, and I would think that you would just scale it up to what you need.
Thank you. So some questions on the Bioflow 720 controller. Is it GMP able with FAT, SAT, IOQ? Is the system 21 CFR compliant? And can data be escalated to a SCADA or Delta V controller?
Yeah, hi, Alwyn. This is Rich. So, yeah, the instrument can be supplied with a full documentation package, including FAT, SAT, IQ, OQ, and then instrument documentation, which could then be used to support implementation into a regulated process. The controller supports 21 CFR Part 11. It allows the local controller to be locked out and kind of driven by a external SCADA, which would be recording and monitoring interaction with the device, as well as creating a batch report, so that data could be used in submission. So was there another part? I'm sorry, I don't have the question in front of me.
The use of SCADA software or Delta V.
Yes, it's compatible with the Eppendorf SCADA platforms, and Delta V will be one of our future releases.
Okay, thank you. Does the ScaleUp Assist function equally for scaling down to smaller sized vessels?
I think this was answered partially in a previous question, but yes, there's no restriction to go in either direction. You could scale up or scale down, just as equivalently.
Thank you. A quick question from how do you control or check for any kind of microbial contamination through the process? Maybe Amanda, that's something you can answer. How do you do it in the lab?
Sure. So there's a couple indicators of a possible contamination. Maybe your gassing, especially in the initial type of like inoculation, your gassing might be very high, and that's unusual typically in the beginning. So that would kind of put up a red flag. DO can drop to 0. If you actually physically take a sample from your bioreactor and look under a microscope, usually you can tell right away. Also, there's usually a little bit discoloration in your culture. It's more like orangey typically, depending on what type of media you're using, of course, but it's more that color. And it certainly, depending how long it's been, it can have a smell as well. So, but I think the best way to check would be actually taking a sample and looking under the microscope for seeing any abnormalities.
All right, another question I think for you, Amanda. What was the volume of the 50 liter sub when it was inoculated?
I believe we put in about, since we ran it at 40 liter max, volume. I believe we put 30 liters in there to begin with, and then adjusted accordingly the media needed after we inoculated. So we inoculated for inoculation density of about 500,000 cells per mil, like I had mentioned. So I can't remember off the top of my head, but I want to say we inoculated about 10 liters of our bioreactor culture, and then it was 40 liter total, so 30 to start.
Okay, thank you. Another question on the ScaleUp Assist software, if it's freely available, and if this software is as well available for non-single-use bioreactors, for example, does keep a BioBlock system?
Freely available, it's included with the instrument. It's part of the integrated software package, so it's a tool integrated in. We are evaluating if that could be added to Eppendorf SCADA platforms or derived in some other way so that it could be used for customers who are not operating a Bioflow 720. And then, as I mentioned earlier, the BioBlue vessel portfolio from the 0.25 up through the Thermo 2000 liter are included, as well as the Bioflow 320 1 liter through 10 liter, and additional vessels will continue to be added. But there are no other vessels, to kind of answer the question, included in that software to date.
Okay, thank you. Two questions on compatibility of the controller to different vessels. So one is about the compatibility to the BioBlue 50C vessel, and the other, if it's able to manage single-use fermentation, such as the 30 or 300 liter from Thermo Fisher.
So the 720 is currently only capable of cell culture applications, but fermentation, and specifically the Thermo Fisher single-use fermentation system, is something we are evaluating for future release, future options, but we do not have it available at launch.
Okay, thank you, Rich. There's a question on, can we show the last barcode again? Amanda, I don't know if you still have the control, if you can show the barcode for the last application note.
There we go.
Thank you. Another question is you use the P over V of 20. Is it possible to enter higher values?
I can answer this one. So yes, the answer is yes. So if you increase in the ScaleUp graph at the end of my portion, I have two graphs. One goes to 0.7 tip speed and the other one to 1.0. So if the customer is able to, their cell line is able to sustain a higher agitation speed, then they can expand the PV values. And also it depends on the vessel. So 10 liter and 50 liter can go more than 50 PV, where the 3 liter is a little bit less. I think it's like 35 or 40 PV. So yes, the 20 is not the upper limit.
Thank you. A question has come in the sub. What was the DO and pH strategy? Amanda, I think that would be good for you.
Okay. So the DO strategy, 50% is usually what we use for all of our CHO cultures. We had done some work with getting the PI values tuned to our process, which we have a ScaleUp, or we have an application for that we can send to you about how to do that. The pH, again, was basically a normal strategy that we've used, pH of 7, that's what our cells like, and we didn't want our deadband to be too high. We were trying to limit any variations of pH, so we kept it at 0.1 deadband.
All right, and what was the highest cell density, and how did you reduce the ammonia levels from increasing as the cells grow?
I guess that one's me. Let me actually go back a graph here. I think everybody's probably done with that. So because they were a batch run, we didn't do anything to, curve the ammonia. The batch run, you don't add anything, so it is what it is type thing. The highest density, I believe, was about 15 million cells per mil, if I remember right. And that was with one of our BioBlue 3C, or 10Cs rather. We got a high density there.
All right. You scaled by P over V. How was the P over V value selected or developed?
I think maybe Rod, you want to take that?
Yeah, I can take this. So, Amanda, if you have control, can you just go back to the end of my session? There's a slide that shows how we actually measured the power numbers. And you'll go back there. And
1 moment, please. This one?
I don't see. Oh, it's not a delay. So,
this one or the previous one?
Yeah, go back a little further to the power number.
Power number, okay.
There you go.
Okay, there you go.
So to measure, to come up with PV, there's a PV equation you can see to the right on the bottom. It says power input. PV equals, power numbers and a bunch of numbers. So this is the equation you would calculate the power input per volume from a power number. And for each vessel, we would have to mechanically determine the power number, as I mentioned briefly during the presentation, using the devices in the picture. You have a torque sensor, flexible adapter, and then there's a protocol that we have released to how to measure the power number. And these devices are custom made. So that's one difficult part, is not everybody has the ability to measure the power number by torque. That's why we measured them for most of our products used in the ScaleUp Assist, and we provide them.
Thank you, Ma. I think we have time for one more question. Are you able to input your own vessel criteria into ScaleUp Assist?
I'll answer that, but perhaps also Amanda, if you could progress to the screen that shows the ScaleUp Assist.
Sure. Hang on one second.
Thanks for your help. But to answer the question, yeah, so where you currently see...
this is from me. Is that okay, or do you want yours?
That's fine.
Okay.
You can see the selection of the various BioBlue and sub-products. Those are actually drop-downs, and one of the available selections in the drop-down is user-defined. And the user-defined, this includes blank fields that you would need to know certain criteria, like a power number, the impeller diameter, and that would then allow you to input your own user-defined and use that in the ScaleUp model if that's not one of the vessels we show in the list.
Okay, thank you. So thank you, Ma, Rich, and Amanda. Any final comments from you for the audience?
Just to thank you for attending today's webinar. We enjoyed presenting it, or I know speaking for my colleagues, we always enjoy the opportunity to present products and the work we do here at Eppendorf.
Yes, thank you guys for participating. And thanks, Alvin, for managing the session. Appreciate it.
No problem. So thank you again for your time today and your important research, Amanda, that you did in the lab. We would also like to thank LabRoots and all involved people at Eppendorf for underwriting underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and their interesting questions that we did not have time for today, and those submitted during the on-demand period will be addressed by one of the speakers or one of the other Eppendorf team members via the contact information you provided at the time of registration. This webinar is part of the online event Streamlining Biologic Scale-Up. We welcome you to stay on the platform. In the auditorium, you will find a panel discussion on the challenges in biologic scale-up. Also, there is an information booth and an applications lab where you can find more information about scale-up applications and our new product, the Bioflow 720. See you there. This webcast can be viewed on demand. Lab booths will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until the next time, and goodbye from me as well.